Here I would like to introduce the rationale and the study design of the TARGET trial. A randomized phase 2 trial of the Trametinib was Pemetrexed in second-line treatment of advanced lung adenocarcinoma with the wild-type EGFR and ALK. On behalf of the lung cancer workshop with the co-chair Professor Mo and Professor Lu and statistician Yan Hong. And Trametinib is a highly selective MET inhibitor, and MET, as a member of the MAPK signal transduction pathway and located in the downstream of the RAF and RAS. And the inhibition of the MET will block the cell proliferation. And as we know that the BRAF and KRAS mutation is high in many cancers, including the melanoma, pancreatic cancer, colorectal cancer, but pretty low in the non-small cell lung cancer. The BRAF and KRAS mutation rate just account for 2% and 5% in non-small cell lung cancer. And the efficacy of the Trametinib in BRAF mutant melanoma is favorable. The response rate is 41%. And another randomized control trial published recently showed that the progression-free survival in the Trametinib group is 4.8 months versus 1.5 months in the chemotherapy group with the hazard ratio 0.45. This is the only data I found in the Trametinib used in the KRAS mutant non-small cell lung cancer. The Trametinib achieved a disease control rate of 64% in KRAS mutant non-small cell lung cancer, in which 5 patients achieved the disease control rate larger than 16 weeks. And in the KRAS mutant non-small cell lung cancer, the median progression-free survival reached 3.8 months, longer than the 2.1 months in the wild-type KRAS mutation. So therefore, the purpose of the trial is to analyze the efficacy, safety, genotyping data for the small molecular MET inhibitor Trametinib in patients with non-small cell lung cancer. Here is the first edition of our study design. The inclusion criteria enrolled patients with advanced adenocarcinoma harboring the BRAF and KRAS mutation received the second-line treatment and randomized 1-to-1 to the Trametinib group and the Pemetrexed group. The primary endpoint is the progression-free survival, and the secondary endpoint is included the response rate, overall survival, and toxicity, and so on. And the patient strategy and stratified by the small status. The sample size, the anticipated sample size is 138 patients based on the progression-free survival 3.8 months versus 2.1 months in the KRAS mutant and the wild-type patients, and using the software pass of the log text. And considering the BRAF mutation rate and KRAS mutation rate is very limited in the Chinese population. And another report showed that there's about 20% of the wild-type BRAF non-small cell lung cancer also achieve a partial response. So the PI considering that there might be the BRAF and KRAS mutation might not be the only strong signal to the MET inhibitor, and therefore they decided to change the inclusion criteria from the target population, just the BRAF mutation and KRAS mutation, to enrich population of the wild-type EGFR and ALK, and which this subset also gained little benefit from the other target therapy. here is our enriched population. And then the study design has changed. Here is the final edition. The patient with advanced adenocarcinoma received second-line therapy with harboring the wild-type EGFR and ALK was included and randomized to the Trametinib group and Pemetrexed group and stratified by the smoking status and biomarker status. In the discussion section, besides the inclusion criteria, there are still many arguments conferring should we choose the phase 2A or 2B design, and should we choose the progression-free survival and overall survival as the primary endpoint. This is my presentation. Thank you for the comment.
Any question?
I have a question. I'm not a statistician, but just use the knowledge what I learned from Dr. Lee's presentation yesterday. I noticed you used an alpha level around 0.5. That means you're going to tolerate a false positive rate around 50%. So what's your consideration?
105. 105. 0.05.
Maybe it's a table?
Oh, P 值0.2. Oh, I missed. It's an error. Sorry.
Thank you.
I have to clarify. It's not Lucian and I to be the PI. We are just the moderator. They are the design. And I'm very impressed with how a discussion will have come to such a very nice presentation in just one overnight. It's very impressive. I really like it. I just want to just make a comment is that throughout the whole discussion is about the molecular target design versus enriched design is always a dilemma. And then we go back and forth. We actually cannot come to any conclusion. But that is a very good exercise process for us to think through. When we try to target a smaller target, what is the best maneuver towards it? But again, excellent job done.
Can I ask a question?
Yes,
The design of using this enrichment runs the risk that your mutants, the BRAF mutant, for example, will be underrepresented because there are a lot more patients who have no BRAF mutation, but ALK and EGFR positive. Is there a way you can ensure that you have sufficient number of BRAF mutations and KRAS mutations in order to actually get a good signal of those groups?
It is a good question, but because the BRAF mutation rate and the KRAS mutation is pretty rare in the total population, and we enrich the population with the wild-type EGFR and ALK, just like this. And in this chart, it will make the percentage of the BRAF and KRAS mutation higher. And in addition, we also have the chance to evaluate the other signal pathway besides the BRAF and KRAS mutation in this subset.
The question is, can you make sure you have a certain slot saved for patients with BRAF and KRAS mutation so you don't lose the signal of those groups? Because otherwise you can overwhelm it with just the patients without the mutations.
Okay, so I also want to commend you and the group for wonderful work, for excellent work in this short period of time. You even come up with the acronym of the study, the target trial, and then you even have a statistician, listed. I'm very happy to see that. And I did check the calculation quickly. I think it's accurate, so that's good. Okay, so I'd like to take this opportunity to make a comment and also kind of bring for some more discussion. That is, this example illustrates very well that although you try to identify the KRAS and or BRAF mutation patient, but because they are so rare, right? So it's not realistic to identify all those patients, patients with all really meet those strict definitions. So you come up with the next best strategy is to have this EGFR wild type and other things, right? And like Lillian said, that even if you do that, there's no guarantee that you will find any KRAS or BRAF mutated patient. Okay, and that's really a challenge for all of us. So I think my comment is that we need to think beyond separate trials, one trial at a time approach. We need to think about a thing called platform approach. And this is similar to what Patty presented earlier. And I think Tony also mentioned about this new design that they are working on in the what is it called? The four. I'm sorry. Focus 4. Okay. So when I hear about this concept of the adaptive design of the Focus 4, my understanding is that you have different. You basically enroll everybody in the sense that and characterize the patient, and depending on the molecular characteristics, then you assign treatment. Okay? And I think we need to think about this kind of approach. That is, we would like to take all comers and then subject them to a molecular testing. Okay, and then depending on outcome, for some patients we know how to treat them. Okay, we assign specific treatment. But for some others, like in this kind of situation, you can identify a subgroup of patients. Then you have some trial for them, and you can do it adaptively. Okay, so that's just my comment. Do you like to think to make additional comments about how do you broaden your scope in terms of making this trial more visible, and also not to lose the patients who does not meet your eligibility criteria?
I totally agree with Jack. Based on our previous design, we will have some risk. We should focus some target. So, but just because we have, based on the previous information, they already signed the agreement, signed the material. So we only this compound versus standard treatment. If we have another another targeted drug compound, we can customize the different style of design trial. You like the battle trials, something we have compared with,
like the adaptive design things. And again, I think we need to think beyond one trial at a time. We need to think about a platform, and we have several trials and for different type of patients.
Yeah, but also like Tony said yesterday, different compound from different company, it's quite complicated.
So I want to the additional comment about all common or the target the population, because I think this is a phase 2 trials. So this phase 2 trials, we want to know the signal for this special population. If the design the phase 3 trials, maybe all common. Okay, we do not lose the time patient, but this is, I think, this is a phase 2 trial.
Tony,
I want to respond to Lian and also the further comment. Agree that in the future that platform design or custody design is probably the way to go, but we have to deal with what we're given. I mean, especially we're giving that piece from the CTAP. That's what we're going to follow. I think it's probably acceptable to use the EGFR mutation L design for one reason, is because we really do not know the efficacy of this drug in the KRAS negative and the BRAF negative patient. In the phase 1 data, there are actually some responder. And although we do not know the molecular profile, and also this is a inhibitor down the MYC and the ELK, which probably will come, a certain kind of like common endpoint or some other kind of pathway. So, although it's not the most so-called specific, but learning something about, the efficacy of this drug in the patient without the KRAS or the BRAF is probably an acceptable maneuver. So I take your point that, we will not have as many as BRAF and KRAS that we like, but we still learn something from this design.
Thank you.